My name is Zavalin. I am a postdoctoral fellow. I recently started working in Caddy's lab, but I've actually known her for years because I did my PhD right next to her lab, and we actually collaborated on a GAT1 project at the time. So I kind of started working with her before I actually joined the lab. So today I'd like to tell you about the GABAergic system and the role of GAT1. So GABAergic signaling mediates most inhibitory neurotransmission in the adult brain, as we've talked about here. It's targeted many drugs with anxiolytic, anti-seizure anesthetic, and sleep-inducing effects. GABAergic deficits underlie neurological and neurodevelopmental disorders, such as epilepsy. And today, I'd like to give a brief overview of the GABAergic system, starting from a relatively basic level for people who are not scientists, with a focus on GAT1. So there are two modes of GABAergic signaling. There's synaptic GABAergic signaling and extrasynaptic. So during a synaptic signal, GABA is released from a presynaptic terminal from one neuron, but it's released up at the top, and it is sensed by the postsynaptic neuron, whereby it activates GABA-A receptors, which are ionotropic receptors. I'll explain what that is in detail in a second. And that leads to a phasic response, as you can see at the bottom, where basically receptors are quickly activated and then they deactivate because GABA is cleared from the synapse. And that's different from extrasynaptic signals, whereby receptors that sit outside the synapse, which include both GABA-A receptors and also GABA-B receptors, which are metabotropic receptors, which act usually more in a modulatory capacity. And these are more sensitive to GABA, and they're activated by a low concentration of GABA that's either a spillover from a large synaptic event, or maybe there's just this presence of a low ambient amount of GABA outside the synapse. And you can see an example of a tonic current down below that's mediated probably by a GABA-A receptor, whereby if you look at that red bar, that's when you administer a blocker of GABA-A receptors. And when you administer that, there's a shift in the baseline, revealing that there was a tonic current present. And at the synapse, you have these, as I mentioned, ionotropic GABA-A receptors, which actually Caddy studied for a long time because mutations in genes that code these are also associated with a number of epilepsies and other neurodevelopmental disorders. And you can see the basic structures. There are the 5 subunits for these receptors. There are a lot of different subunits they can form, about 18 different ones, typically 2 alpha, 2 beta, and a gamma or a delta. And that's actually kind of important because depending on whether the gamma or the delta are present in that receptor, they can localize either to the synapse or outside the synapse. So typically, the delta-containing ones are outside the synapse and extrasynaptic, and then the gamma-2 containing ones are at the synapse. And as I mentioned, these receptors are ionotropic, meaning that they are ion channels, so they conduct chloride. And so GABA will bind this receptor, will open, and chloride can go either way. So typically in a mature neuron in the adult brain, what happens is when these receptors open, as you can see there on the right, the chloride concentration is a lot higher outside the neuron. It's very low in the neuron. So when the receptors open, chloride rushes in, and that's an inhibitory response. And that is due to activity of a transporter called KCC2, which will constantly extrude chloride from the neuron. But during early development or in certain pathological conditions, the activity and expression of KCC2 is pretty reduced, and then activity of a different transporter, NKCC1, is high, as you can see there on the left in green. And that actually will pump chloride into the neuron. So during development, GABA actually is not an inhibitory signal, it's an excitatory signal, and that's very important to promote a whole number of different developmental events. We don't have time to go into a lot of detail here, but it's actually a fascinating field of its own. I actually used to study this as part of my PhD, and we have several experts on this at Vanderbilt, including Dr. Eric Delpierre. And this is also something to consider either when treating really young patients where potentiating the GABAergic system can have a different effect than the inhibition you would expect at a later developmental stage. And also there can be abnormalities in this associated with different disorders that can lead to GABA losing its inhibitory effect. It's also important to consider. Some of the other important players in GABAergic signaling are the GAD enzymes, so GAD65 and 67. They're shown here in that dark blue presynaptic terminal at the top. So basically that's the enzyme that's going to convert the glutamate to GABA. And then VGAT is what loads GABA into synaptic vesicles. And then GABA is released, and then there are GABA transporters, GAT1 and GAT3, and these are going to take GABA back up into neurons and into astrocytes, and there's an astrocyte shown here in light blue. And there are a number of drugs that will target different components here, all with the effect of potentiating GABAergic signaling and relieving seizures. And today, of course, I'd like to focus on GAT1, and I'll mention very briefly GAT3 as well. So the structure of both GAT1 and GAT3 is that they're a dimer, so two proteins will come together. You can see the basic structure of a single subunit there on the right. There are 12 transmembrane domains. And basically the way they work is they use the sodium gradient. There's a lot more sodium outside the neuron, and they use that to transport GABA into the neuron. So 2 sodium, 1 chloride, and then a GABA molecule will be transported into the neuron during one transport cycle. This is some data from our lab. So I mentioned there are 2 different GABA transporters, GAT1 and GAT3. So GAT1 is expressed by Western blot, you can see in many of the tissues, so in cortex and cerebellum, hippocampus, thalamus, both in neocortex and other structures, expressed quite highly. GAT3, on the other hand, is expressed quite robustly in thalamus, but not very highly in some of these other regions, like the neocortex. And so this is data from our lab that Dr. Wang Zhangshen got. And then this here is data from a paper previously published by other authors showing immuno. And again, you can see here that GAT1 expression is very high throughout the CNS. So you see it in cortex, and striatum, hippocampus, thalamus. GAT3 is a lot more restricted, mainly in thalamus. So I think this just shows just how important GAT1 is, because it's very much the primary mode of GABA reuptake in many of these brain regions. As some of the presenters mentioned today, a GAT1 knockout mouse has been present in the field for a while, and that tells us a lot about how important GAT1 is for brain function. So at first it was published in 2003, generated by Istvan Modi's research group. And this mouse has been shown to have absence seizures, as actually Dr. Gray very well has shown us today. But also this has been mentioned in the Cope et al. Paper. It's also sensitive to induced seizures. It has motor disorders, a number of different motor disorders, including that rotarod test that was mentioned previously that it does not do very well on. It may have mild anxiety, but the studies on this seem to be conflicting, and it has certain cognitive deficits. But I personally am an electrophysiologist, and that's what I'm going to talk about for the rest of this talk. And so one of the things that was observed in this mouse was an increased tonic GABAergic current. So again, as I showed you at the very beginning, that would be that activation of those extrasynaptic receptors. So in the image deck, similarly to what I showed you, in this study, they basically just applied a drug that blocks GABA-A receptors, and they saw a shift in the baseline for these neurons. So in this study, they looked at hippocampal neurons, and they saw, if you look at what, I don't know, those numbers might be too small, but basically in the knockout, they saw 45 picoamp current. And in the wild type, it was a 5 picoamp current, basically a current, there was really no current. So suddenly this large tonic current has emerged, likely due to all this ambient GABA being present outside the synapse. This has also been observed in thalamus. So thalamus, even in wild-type animals, has a pretty large resting tonic current. So it's about 40, 50 picoamps. But in the knockout, you can see it's pretty much doubled here. And in cortex, cortex usually does not have very much of a tonic current, but in the knockout, about a 10 picoamp current was observed. In this paper, the Bragen et al. 2008, they also did microdialysis. So this is measuring GABA levels in the cortex of these mice. They saw perhaps an elevation, but nothing significant at baseline. But when they basically elicited a very large release of GABA by washing on potassium, suddenly they saw this very large spike in the knockout animal. And you can see that spike. Those are like the dark, the gray circles. The spike was much higher in the knockouts than in the wild-type mice, suggesting there's an issue in clearing this surge of GABA. These studies also looked at synaptic events, so those would be like those quick phasic events that happen at the synapse that I showed you earlier. They looked at them at hippocampus in the Jensen et al. Study. They didn't see a difference either in the kinetics, so kind of the shape of each of the event, nor in the frequency of events, so how many of these events occur per period of time. But then they looked at miniature events, and these events differ because a specific drug called tetrodotoxin is washed on, and when that wash-on happens, all spontaneous activity, that is a neuron actually being excited and releasing GABA to send a signal, that stopped. So only fusion that just happens on its own without a neuron signaling occurs. And at this time, they actually saw that there were significantly fewer events in the GAT1 knockout, and they hypothesized this was due to a GABA-B mechanism whereby the release of synaptic vesicles was modulated. And the Bragen et al. Study that also looked at spontaneous IPACs and also didn't see a change, although unlike the previous study, this one was in cortex. They didn't see a change in kinetics or frequency, but then what they did is they looked at evoked events, and this is where instead of measuring a single synaptic events that occur kind of on their own, they would record events from a layer 2 neuron and put a big stimulating electrode in layer 4 and cause a very large event to occur, which probably involved multiple synapses. And at this point, when there was a much larger event, a lot more GABA involved, you could see a significant difference between the knockout and the wild type, where, as you can see, the knockout has this prolonged decay. It takes a lot longer for all that GABA that was released to be cleared from the synapse, so the receptors are activated for a longer period of time. So I think it makes sense then that so many mutant variants of SLC6A1, which codes for GAT1, are pathogenic, considering how important GAT1 is, both on a physiological level and on the level of looking at what happens to the entire animal. In our lab, as Caddy has previously alluded, we are studying two knock-in mouse models, one for S295L and one for AA288V. We've, I think, talked enough about both of these mutations, so I won't go into detail about those, but I will mention that in these model mice, we have absence seizures and reduced motor activity. And unlike the GAT1, so the GAT1 knockouts, that is a homozygous mouse. In these models, at least as far as the seizures are concerned, these are heterozygotes, so they're still expressing one wild-type allele, and they're still showing absence seizures similar to the patients. In both of these models, we see that there is reduced expression of GAT1. So here I'm showing you both heterozygote mice and homozygote mice. So if you look at homozygote mice, so the top is the A288V and the bottom is the S295L. So you can see that for A288V, for the homozygote mice, only about 25% of GAT1 is being expressed. And for the S295L, there's pretty much no GAT1 being expressed, it's all being degraded. And if you look at the heterozygous expression of both of them, you can see about a 50% decrease. So basically like one copy, the wild-type copy, is still working. Yeah, so for A288V, there's basically partial loss of function because of this, and S295L has complete loss of function. And when you measure GABA uptake, basically in this case, what we did is we took cultured HEK cells and we would transfect them with the different combinations of GAT1, so either like a wild-type GAT1 or some of these mutant variants, and we looked at how well these cells would take up radioactively labeled GABA. So if you do that, you can see that if you only express wild-type, they can take up GABA, so that's the bar on the very left, and it's normalized to be 1. And then the S295L, basically, if you only express that, they can't take up any GABA. If you express, that's on the very right, if you express A288V, it's about 75% reduction in how much they can take up GABA. And then if you express like 50% wild type, 50% one of the different variants, you can see that still the uptake is significantly reduced from a complete wild type expression. And we also did this experiment in synaptosomes. So these, basically what this is we'll dissect brains out of these knock-in mice, and then we'll isolate synaptosomes. So these are basically, you basically isolate synapses is what you're doing here. And then you look at how well this can take up GABA as a measure of a more in vivo relevant, at least, model than HEK cells. So in this case, what we saw was, again, a significant decrease in heterozygotes to uptake GABA into the synaptosomes. So both S295L and A288V have significantly reduced uptake. And what we looked at next, and this is the research I've done in the lab so far, is we looked at the physiological implications on the neuronal level of both the reduced uptake and reduced trafficking. So what you can see here is, first I looked in cortical neurons. So this is layer 6 somatosensory cortex. And that's somewhat relevant because I was comparing my research to Dr. Kavanaugh's research, for instance, and maybe he'll talk about it later. So comparing a homozygote S295L to a wild type, I didn't see a difference in the kinetics, in the amplitude or in decay. But what I am seeing so far is there is a change in the inter-event interval. So the events are less frequent, so you have fewer synaptic events in these homozygotes. And another region I looked at was in thalamus, and in thalamus I compared homozygote to a heterozygote. And in this case, I actually didn't see a difference, neither in amplitude nor in decay nor in frequency. So that looked normal, although perhaps if I look at a homozygote, then I would actually see some sort of a difference. I also looked at tonic currents, and I looked in the cortex, where there's typically no tonic current present. And I actually, in the homozygote, I did not observe a tonic current. So at 2-3 months old, I measured a pretty significant number of cells. I didn't see a significant current in the homozygous mice. I also looked at a later time point. Some of the data in our lab suggested that as these mice get older, they perhaps then start exhibiting a tonic current. And we thought that this would emerge if we looked at older mice. So I looked at heterozygote and homozygote S295L mice, again measuring a tonic current in the cortex. And I still have not picked up a significant tonic current there. And I looked at the tonic current in thalamus. And thalamus, as I mentioned earlier, has a baseline tonic current that's pretty high, even in the wild type. And that's what I saw. I saw about a 30 picoamp current in the wild type mouse. And when I compared that to a heterozygote S295L mouse, I surprisingly did not see a difference here in this region. So to summarize what I've told you so far, GAT1 is one of the primary GABA transporters in the CNS, and GAT1 knockout mice have spontaneous absence seizures. They have increased tonic GABAergic current and some alterations in synaptic events. And in the A288V and S295L knock-in mice that we study, that are based on dominant patient variants, they have spontaneous absence seizures in heterozygotes, reduced GAT1 expression, reduced GABA uptake. They have normal tonic currents in ventrobasal thalamus and cortex, and normal amplitude of thalamic and cortical synaptic events in the ways that I've measured them so far. But they have reduced frequency of spontaneous iPSCs in cortex. So what are future directions? So for me, as a physiologist, I'd like to know how GABAergic neurotransmission is altered in both of these knock-in mice. As I think studies with the knockout mice have shown, you won't find a deficit in every way that you look when you're looking at these synaptic currents. You have to look at a specific age or a specific instance, either when there's a strong surge of GABA or in other similar situations. So that's what I'm doing so far. One of the experiments I'm doing, as I'm showing here, is I'm looking at evoked currents where I'm stimulating layer 4, recording from layer 2, and then measuring basically response to a big surge of GABA. So I'm really hoping to find a difference there. So I'd like to thank you very much for your time, and to thank my lab members here. And I'll let the next speaker talk.